breaking the code, new molecular targets reset hallmarks of aging. Welcome to the official GlobalRPh YouTube channel. If you haven't already, hit that subscribe button to stay updated with our latest videos. Researchers at the University of Texas MD Anderson Cancer Center have recently broken new ground in the hallmark of aging research by demonstrating that restoring youthful levels of telomerase reverse transcriptase, TERT, can substantially reduce aging signs and symptoms in preclinical models. This groundbreaking work represents a potential turning point in our understanding of how molecular targets can reset biological aging processes. The hallmarks of aging encompass a set of interconnected biological mechanisms that characterize the aging process across different organisms. Initially identified as 9 hallmarks of aging with special emphasis on mammalian aging, researchers have now expanded this framework to 12 distinct hallmarks: genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, disabled macroautophagy, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, altered intercellular communication, chronic inflammation, and dysbiosis. These hallmarks form the foundation of modern cellular aging theory and provide specific targets for intervention. Furthermore, the therapeutic restoration of TERT levels has shown remarkable effects, including new neuron formation in the hippocampus, improved cognitive performance, and reduced inflammaging, an age-related increase in inflammatory markers associated with multiple diseases. These findings may have profound therapeutic implications for age-related diseases such as Alzheimer's, Parkinson's, heart disease, and cancer. The discovery that aging rates can be controlled by genetic pathways and biochemical processes conserved throughout evolution has transformed aging research. Throughout this video, we will examine the molecular triggers behind the hallmarks of aging, explore emerging therapeutic targets that reset aging pathways, analyze their interconnected effects, and discuss the translational potential of these interventions in clinical settings. Molecular triggers behind the hallmarks of aging. The progressive deterioration of cellular function with age stems from fundamental molecular changes occurring at the genomic level. These alterations form the basis of the hallmarks of aging theory, providing critical insight into why organisms experience functional decline over time. Genomic instability and DNA repair failure. Living organisms continuously face DNA damage from both exogenous and endogenous sources. Each day, human cells endure approximately 10,000 abasic sites and thousands of oxidative lesions. Accordingly, cells have evolved sophisticated DNA repair mechanisms: base excision repair, BER, nucleotide excision repair, NER, mismatch repair, MMR, homologous recombination, HR, and non-homologous end joining, NHEJ, to maintain genomic integrity. However, the efficiency of these repair mechanisms declines with age, leading to the accumulation of unrepaired DNA damage. This accumulation of somatic mutations across the lifespan has been identified as a dominant driver of aging. Telomere attrition and replicative senescence. Telomeres, the protective DNA protein structures at chromosome ends, naturally shorten with each cell division due to the end replication problem. Human liver tissues lose approximately 55 base pairs of telomeric DNA annually. When telomeres reach a critical length, cells enter senescence or undergo apoptosis. This telomere-driven, replicative senescence represents a powerful tumor suppressive mechanism, but simultaneously contributes to aging. Notably, critically shortened telomeres trigger a persistent DNA damage response that activates p53 and inhibits the PGC1-alpha pathway, consequently impairing mitochondrial function. Epigenetic drift and chromatin remodeling. Age-related epigenetic alterations include global DNA hypomethylation alongside site-specific hypermethylation, particularly at CPG islands. Additionally, histone modifications undergo significant changes: increased H3K4 trimethylation and H4K16 acetylation, alongside decreased H3K9 and H3K27 methylation. One early model of aging, the heterochromatin loss model, suggests that heterochromatin domains established during embryogenesis break down with age, disrupting nuclear architecture and gene expression patterns. This loss of heterochromatin is evident in senescent cells through decreased H3K9Me3 levels. Loss of proteostasis in aging cells. Protein homeostasis, proteostasis, relies on a complex network of molecular chaperones and proteolytic machinery that coordinates protein synthesis, folding, and degradation. With age, the activities of the two principal proteolytic systems, the autophagy lysosomal system and the ubiquitin proteasome system, decline markedly. This decline leads to the accumulation of misfolded, oxidized, and damaged proteins. The loss of proteostasis particularly affects post-mitotic cells like neurons, which cannot dilute damaged proteins through cell division, ultimately contributing to age-related neurodegenerative diseases. New molecular targets that reset aging pathways. Recent advances in aging research have identified molecular targets that can reset multiple hallmarks of aging. These emerging interventions demonstrate remarkable potential for reversing age-related cellular dysfunction. TERT reactivation and telomere elongation. Scientists recently discovered a small molecule TERT activating compound, TAC, that epigenetically de-represses the TERT gene, restoring youthful telomerase expression levels. In aged preclinical models, TAC treatment led to new neuron formation in the hippocampus while improving cognitive performance. Moreover, TAC treatment reduced inflammaging in both blood and tissue samples while eliminating senescent cells through p16 gene repression. The compound works by upregulating TERT transcription via the MEK-ERK-AP1 cascade, promoting telomere synthesis and silencing p16-INK4A expression through DNMT3B-mediated promoter hypermethylation. Remarkably, TERT functions beyond telomere extension by acting as a transcription factor, affecting genes involved in neurogenesis, learning, and inflammation. SIRT6 and chromatin stability restoration. SIRT6, a chromatin-bound enzyme, emerges as one of the earliest responders to DNA damage, reaching damaged sites within approximately 5 seconds. This histone deacetylase coordinates proper DNA repair through a complex response including recruitment of the ATP-dependent chromatin remodeler SNF2H to DNA break sites and rapid histone H3K56 deacetylation. Through these mechanisms, SIRT6 influences recruitment of downstream repair factors such as 5,3-BP1, gamma-H2AX, RPA, and BRCA1. Beyond DNA repair, SIRT6 has been implicated in telomeric chromatin maintenance, co-localizing with HP1 beta within the nucleus. mTOR inhibition and autophagy induction. Rapamycin, an mTORC1 inhibitor, remains the only known pharmacological treatment increasing lifespan across all model organisms studied. While rapamycin's side effects include immunosuppression and metabolic disturbances, these appear primarily mediated by mTORC2 inhibition, whereas its beneficial effects on healthspan stem from mTORC1 inhibition. Importantly, intermittent or low-dose mTOR inhibitor regimens may provide geroprotection with reduced side effects. The primary mechanism behind rapamycin's benefits involves autophagy induction, a process whereby cells recycle damaged proteins and organelles. With age, autophagy becomes impaired in multiple mouse cell types, yet genetic autophagy activation rejuvenates aged mouse livers histologically and functionally. NAD+ boosters and mitochondrial repair. NAD+ precursors like nicotinamide riboside (NR) demonstrate profound rejuvenating effects. NR treatment increases NAD+ concentration in muscle stem cells from both young and old mice. This intervention enhances mitochondrial function, activates mitophagy, recycling of dysfunctional mitochondria, and improves stem cell regenerative capacity. Additionally, NAD+ restoration leads to stem cell rejuvenation and improved oocyte quality in aged mice. The mechanism involves NAD+'s role as a cofactor for sirtuins, particularly SIRT3, which regulate mitochondrial homeostasis through deacetylation and activation of stress responder FOXO3 alpha. Interconnected effects on the 9 hallmarks of aging. The intricate interplay between hallmarks of aging reveals how targeted interventions can trigger cascading effects across multiple cellular systems. Experimental evidence demonstrates that modulating one specific hallmark typically influences several others, underscoring the complex nature of aging as an integrated process rather than isolated deterioration pathways. Impact on cellular senescence and SASP. Senescent cells secrete a diverse array of molecules, collectively known as the senescence-associated secretory phenotype, SASP. This includes inflammatory cytokines, chemokines, growth factors, and matrix remodeling enzymes that profoundly alter the tissue microenvironment. Interleukin-6, IL-6, stands out as the most prominent SASP cytokine, accompanied by IL-1, alpha, beta, and chemokines such as IL-8, CXCL8. These factors operate through both autocrine and paracrine mechanisms, reinforcing senescence in the secreting cells themselves and inducing senescence in neighboring cells. Though originally considered tumor suppressive, SASP exhibits context-dependent effects, sometimes promoting tumor development in breast, skin, prostate, and pancreatic tissues. Stem cell exhaustion reversal via FOXO3. FOXO3, a forkhead transcription factor, plays a crucial role in preserving stem cell populations across different tissues. In neural stem cells, FOXO3 prevents premature depletion by maintaining self-renewal capacity and differentiation potential. Similarly, in hematopoietic stem cells, FOXO3 deficiency leads to increased ROS levels, defective quiescence maintenance, and heightened sensitivity to myelotoxic injury. Essentially, FOXO3 coordinates quiescence, stress resistance, and glucose metabolism in adult stem cells, functions critical for preserving stemness throughout the lifespan. Improved intercellular communication through cytokine modulation. Age-related alterations in intercellular signaling represent another hallmark that intersects with multiple aging processes. Blood factors from older organisms can induce aging signatures in younger ones, highlighting how circulating factors influence aging. SASP factors actively reshape the microenvironment by recruiting immune cells, altering vascular function, and affecting neighboring cell behavior. For instance, senescent cells interact with platelets through SASP, increasing platelet aggregation and promoting endothelial dysfunction. Reduced chronic inflammation via NF-kappa B suppression. NF-kappa B emerges as a central regulator connecting inflammation to multiple hallmarks of aging. This transcription factor drives expression of pro-inflammatory cytokines, TNF-alpha, IL-1, IL-6, chemokines, and enzymes like INOS and COX-2. While essential for acute immune responses, persistent NF-kappa B activation leads to chronic inflammation and contributes to rheumatoid arthritis, inflammatory bowel disease, atherosclerosis, and neurodegenerative conditions. Notably, interventions targeting TNF-alpha with etanercept from 16 to 18 months in C57BL6 mice prevented sarcopenia and increased lifespan. Therapeutic implications and translational research. Translating theoretical understanding of aging mechanisms into clinical interventions represents a critical frontier in geroscience. These emerging therapeutic approaches target specific hallmarks of aging at the molecular level. Senolytics targeting P16-INK4A positive cells. Selective elimination of senescent cells offers remarkable therapeutic potential. Studies demonstrate that removing P16-INK4A positive cells delays onset of age-related pathologies in tissues, including adipose, skeletal muscle, and eye. Specifically, late-life clearance attenuates progression of already established age-related disorders. Various elimination approaches show consistent benefits. Targeted apoptosis restores tissue homeostasis in response to chemotoxicity. Directed elimination via BCL-W and BCL-XL inhibition improves physical function. and clearance of p16-positive cells increases lifespan in aged mice. Epigenetic reprogramming using Yamanaka factors. Artificial cell reprogramming with transient Yamanaka factors offers rejuvenation without pluripotency risks. Cyclic administration, 2-day pulse, 5-day chase, of OSKM factors in progeric mice increased median lifespan by 33%. Even more striking, delivering OSK, excluding CMYC, via AAV9 to 124-week-old mice extended remaining lifespan by 109%. Chemical alternatives like VC6TF cocktail show similar rejuvenation effects without genetic manipulation. Caloric restriction mimetics, metformin and rapamycin. Both metformin and rapamycin mimic caloric restriction benefits through mTOR inhibition. Rapamycin extends lifespan across multiple species, yet presents side effects including immunosuppression. Meanwhile, metformin activates AMPK, increasing insulin sensitivity while lowering hepatic gluconeogenesis. Although metformin is generally well tolerated, gastrointestinal side effects such as nausea, diarrhea, and abdominal discomfort occur in approximately 20 to 30% of patients, particularly during dose escalation. Gene editing tools for age-related mutation correction. CRISPR-Cas9 technologies offer precise genetic correction for age-related diseases. Recent applications include correcting PSEN1/PSEN2 mutations in Alzheimer's iPSCs, SOD1/FUS mutations in ALS, and using ABE-Max VRQR to correct approximately 90% of LMNA mutations in HGPS patient cells. Despite therapeutic promise, challenges remain regarding delivery methods, off-target effects, and potential immunogenicity. Conclusion. Research into the molecular mechanisms of aging has advanced remarkably over the past decade, shifting from mere understanding to actual intervention. The 12 hallmarks of aging now provide a comprehensive framework for targeting the fundamental processes that drive biological decline. Researchers have demonstrated that these hallmarks do not operate in isolation, but rather form an interconnected network of cellular and molecular pathways. Telomerase reverse transcriptase, or TERT reactivation, stands as a particularly promising approach, demonstrating profound effects on neurogenesis, cognitive function, and inflammation reduction. Beyond telomere extension, TERT's role as a transcription factor expands its therapeutic potential across multiple aging hallmarks. Similarly, SIRT6 restoration addresses chromatin stability and DNA repair mechanisms that deteriorate with age, while mTOR inhibition through compounds like rapamycin enhances autophagy and cellular recycling processes essential for maintaining proteostasis. The ability to target one hallmark and subsequently affect others highlights the integrated nature of aging biology. Cellular senescence and the associated SASP clearly exemplify this interconnection, as interventions that reduce senescent cell burden simultaneously mitigate inflammation, improve stem cell function, and enhance intracellular communication. FOXO3 activation emerges as another critical node in this network, preserving stem cell populations across different tissues and preventing premature exhaustion. Translational efforts have already yielded several promising therapeutic strategies. Senolytics specifically target P16-INK4A positive cells, effectively removing senescent cells that contribute to age-related pathologies. Epigenetic reprogramming using Yamanaka factors represents another groundbreaking approach capable of resetting cellular age without risking uncontrolled growth. Additionally, caloric restriction mimetics such as metformin and rapamycin offer practical interventions that target nutrient-sensing pathways implicated in aging. Gene editing technologies present perhaps the most precise approach for addressing age-related mutations, though challenges regarding delivery methods and safety remain. These various interventions collectively demonstrate that aging processes can be slowed, halted, or even reversed through targeted molecular approaches. Future research will undoubtedly focus on optimizing these interventions while minimizing side effects. The field must also address questions regarding timing, dosage, and combination therapies to maximize healthspan extension. Despite these challenges, the foundation has been laid for a new era in medicine, one where age-related diseases might be addressed not merely as individual conditions, but as manifestations of underlying aging processes amenable to intervention. The capacity to reset multiple hallmarks of aging through defined molecular targets represents a paradigm shift in how researchers approach age-related decline. This evolving understanding promises to transform clinical practice from disease-centric treatments toward comprehensive interventions that address the fundamental causes of aging itself. Though the path toward clinical implementation remains complex, the extraordinary progress detailed throughout this video suggests that meaningful extension of healthy human lifespan may become achievable within the foreseeable future. Key takeaways. Scientists have identified molecular targets that can reset aging processes, offering unprecedented opportunities to extend healthy human lifespan through targeted interventions. TERT reactivation reverses multiple aging signs. Restoring telomerase levels promotes new neuron formation, improves cognitive function, and reduces age-related inflammation. Aging hallmarks are interconnected networks. Targeting one mechanism like cellular senescence simultaneously affects inflammation, stem cell function, and intracellular communication. Senolytics eliminate harmful senescent cells. Removing p16-4A positive cells delays age-related diseases and restores tissue homeostasis in multiple organs. Epigenetic reprogramming resets cellular age. Yamanaka factors can rejuvenate cells without cancer risks, extending lifespan by over 100% in aged mice. mTOR inhibition mimics caloric restriction benefits. Compounds like rapamycin enhance autophagy and cellular recycling, extending lifespan across all studied species. These breakthrough discoveries represent a paradigm shift from treating individual age-related diseases to addressing the fundamental molecular causes of aging itself. The convergence of multiple therapeutic approaches, from gene editing to senolytic drugs, suggests that meaningful health span extension may become clinically achievable in the near future. Frequently asked questions. Question 1. What are the key hallmarks of aging? Scientists have identified 12 distinct hallmarks of aging, including genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, and cellular senescence. These hallmarks form an interconnected network of biological mechanisms that characterize the aging process across different organisms. Question 2. How does TERT reactivation impact aging? TERT reactivation has shown remarkable effects on reversing aging signs. It promotes new neuron formation in the hippocampus, improves cognitive performance, and reduces age-related inflammation. TERT also functions as a transcription factor, affecting genes involved in neurogenesis, learning, and inflammation. Question 3. What are senolytics and how do they work? Senolytics are therapeutic compounds that selectively eliminate senescent cells in the body. By targeting p16-INK4A positive cells, senolytics can delay the onset of age-related pathologies in various tissues, including adipose, skeletal muscle, and eye. This approach has shown promise in restoring tissue homeostasis and improving physical function in aged organisms. Question 4. Can epigenetic reprogramming reverse aging? Yes. Epigenetic reprogramming using Yamanaka factors has shown potential in reversing cellular age. Partial reprogramming, particularly using the OSK factors, has demonstrated significant life extension in aged mice without the risks associated with full cellular reprogramming. This approach offers a way to rejuvenate cells and tissues without inducing pluripotency. Question 5. How do caloric restriction mimetics like metformin and rapamycin affect aging? Caloric restriction mimetics, such as metformin and rapamycin, work by inhibiting the mTOR pathway, which is involved in nutrient sensing and cellular metabolism. These compounds can extend lifespan across multiple species by enhancing autophagy, improving cellular recycling processes, and maintaining proteostasis. However, they may also have side effects that need to be carefully managed in clinical applications. Thanks for watching our GlobalRPh production. If you enjoyed the video and found it helpful, don't forget to smash that like button and subscribe to our brand new YouTube channel.